My MiCM summer project was about integrating machine learning applied to risk prediction of stress-related mental disorders. The goal of this project was to identify genetic and psychosocial factors associated with major depressive disorder, or MDD. We had data from a cohort of 1,403 participants, and the data included information on single nucleotide polymorphisms, or SNPs, and psychosocial variables from a questionnaire that the participants completed. These included information about MDD in lifetime, MDD in the last year, childhood trauma, drug or alcohol problems, and many others. We first performed association analyses between the SNPs and the MDD phenotypes we had. We didn't find any significantly associated SNPs, and we thought maybe this was because our cohort was relatively small, since we only had about 1,400 participants. So next, we turned to the psychosocial data we had from the questionnaire. We wanted to predict whether a participant had MDD or not based on the other questionnaire variables. We used a model called MiCM, which is a Bayesian latent topic model initially designed for modeling electronic health record data. The model is unsupervised, and the way it works is that we tell it to learn a fixed number of latent topics or metaphenotypes from the training data. For each metaphenotype, we have a membership value for each of the original features, and this gives us a probabilistic interpretation of the metaphenotypes. We use this model on only questionnaire data first after removing MDD variables from the training data. We trained MiCM for 30 metaphenotypes, and we use these metaphenotypes to train a logistic regression model to predict if the participant had MDD or not. Here are the classification results we obtained using 5-fold cross-validation. We can see that MiCM produces similar results as principal component analysis with 30 components, and both perform a lot better than when we only use the raw features as input to the classifier. But one advantage of MiCM over PCA is that it assigns a membership value for each feature, for each metaphenotype, so we have a probabilistic interpretation of the output. For example, here we see the top 5, on the left, we see the top 5 questionnaire variables for the 10 metaphenotypes that were the most correlated with MDD. And on the right, we have all of the 30 metaphenotypes sorted by decreasing correlation with MDD, and we can easily see which of our original features have a high membership value for these metaphenotypes. We next try to jointly model the questionnaire data and the SNP data. Here, the MiCM results are worse than if we use just the raw features or if you use PCA. We think this might be because we have too many SNPs. We had about 100,000 SNPs after quality control and pruning, and we only had about 400 questionnaire variables. So there's an imbalance between our two types of data. And we're not entirely done with this project yet. For the next step, instead of using SNPs, we're going to try using gene expression data from different brain tissues. We can derive those from the SNP data. And this way, we would only be dealing with a few thousand genes instead of 100,000 SNPs. Another analysis we plan on doing is to train MiCM on all the phenotypes, including the MDD phenotypes, then performing association analyses on the SNPs. This could help the problem we initially had related to lack of statistical power. Thank you.